back. So this is biology question 44 from IMAT 2011 past paper. So I will be answering this question. So it says, which of the features below may be present in both prokaryotic cells and eukaryotic animal cells? Okay, so we are given with four lists of these structures, and we must find out which of these four structures are found in both prokaryotics and eukaryotic animal cells. Okay, so before we move on forward, we must recall the basic definition about the prokaryotics and eukaryotics. Okay, so basically, what's the main features that we must know is that prokaryotic organism like bacteria, archaea, they all uh, lack membrane, membrane bound organelles. Okay, so the most of the organelles that you might see in the prokaryotes, they all lack membrane bound. So there's almost very little or no membrane bound organelles. And whereas the eukaryotic, they have a lot, extensive amount of membrane bound organelles. Okay, so there's one thing you must be clear that this is about organelles. Like for example, in the eukaryote, there's a nucleus. Okay, there's a chloroplast. There can be mitochondria, mitochondria, and there's ER and etc. So these are like the membrane-bound organelles. Okay. Okay, I mentioned chloroplast here because I'm generally explaining about eukaryotic because it's not just animal cell. Eukaryotic can also be a plant cell, it could be a fungi or it could be yeast as well. Okay, so there's many of them apart from animal cells, so I'm just being generalized. Okay, so eukaryotic organisms have extensive amount of membrane-bound organelles to help carry out more complex function, but prokaryotic lack membrane-bound organelles. Okay, so that's the difference, and that we must know about it. Okay, so prokaryotic lack membrane-bound organelles, so that's the difference you must know. And the second one is that most of the almost all every prokaryotic organism are single cell organism. Okay, so all prokaryotic organism, including bacteria, archaea, are single cell. But whereas eukaryotic, they are multicellular. Some are multicellular, and are some are single cell. So the single cell, the best example I can give is the yeast cell. So yeast cell are eukaryotic organism because they contain membrane-bound organelle. They have nucleus to enclose their genetic materials and etc. So that's why yeast cell I can give the best example to be eukaryotic, and multicellular like animals, plants. Those are the multicellular organism. Okay, so that's the main thing you must know. And what else you must know? Yeah, so the thing is, the prokaryotic, most of them have a cell wall. Okay, so most of them have cell wall, but and also the eukaryotic organism has a cell wall, like it is a fungi and the plant cells. Okay, so now again, I'm being very general about eukaryotic and prokaryotic cells. So they have mentioned animal cell. Animal cell do not have cell wall, but I'm being generalized about eukaryotic. Okay, so both has the cell wall that we must know, and also uh, they both of them also has the ribosomes. Both has ribosome because that's important for protein synthesis. Okay, so that's the very general idea about the prokaryotics that you must know. Okay, so now I'm going to forward about four of these structure and list out the basic function and their definition. Okay, so first of all, we'll go with glycogen. So the main What's glycogen is basically it's a highly branched, highly branched polymer of alpha glucose. So it's a highly branched polymer of alpha glucose. So alpha glucose are like a monomer units. They join in together to form this glycogen. And most importantly, each of the alpha glucose are glycosidic bonded. So they're 1,4 and 1,6. So this are the glycosidic bond. Okay, so this makes a very branched structure because we know glycogen is not a linear structure, it is a highly branched structure. Okay, so there also the main function is that glycogen, their main, okay, so their main form of glucose storage. So glucose we know is the main compound that are required to produce energy, which is ATP. So glucose are broken down into ATP. So this is main source of fuel for our body, for most of the organisms. So their main form of glucose storage. And right down here, I'm going to write the things that are common and where are they found. Okay, prokaryote and eukaryote. And since the question mentions animal cell, or here, animal cell, so like this, so animal cell. Okay, so I am going to be now strict about animal cell. Okay, but since the prokaryotic organism, they haven't specified any sort of cell because we know prokaryotic are divided into two domains. It is bacteria and archaea. But for your ease, I'm going to just write a basic example like bacteria. Okay, so okay, so as we were discussing about the glycogen, so glycogen are found in both. So in what forms they're found are quite different. That makes it different for purpose. Okay, so glycogen, they are common in both prokaryotes and eukaryotes, but they exist in different forms. So the glycogen in prokaryotes exist in form of small granular glycogen. Okay, small granular glycogen. So in the cytosol, which is also called cytoplasm, whichever way you like. But to be more specific, I'll say cytosol. Okay, so glycogen exists in form of small granular glycogen in the cytosol of prokaryotes because you know they lack membrane bound organelles, so they almost every structure are free floating around the cytoplasm. And for the eukaryotes, the glycogen are located in skeletal muscle, skeletal muscles, and most importantly in the liver and in liver. So these are the locations where the glycogen are located in the eukaryotes and the prokaryotes. And they exist in different forms. So for prokaryotes, they generally are in the form of small granular glycogen. Okay, so for eukaryotes, the glycogen are quite large in structure, but they're located in skeletal muscle and liver. Okay, so we can mark glycogen to be common in both prokaryotes and eukaryotes. Now moving on forward with the cell wall. So as I've mentioned before, the cell wall do exist in eukaryotic, but since the question focuses about animal cell, we can easily pinpoint this out as wrong. But I'm still going to explain very general function and the purpose of the cell wall in the organism like plants and bacteria. Okay, so first of all, it's a rigid cell wall, a rigid layer of polysaccharide, polysaccharide that are located outside outside of plasma membrane. Plasma membrane is the outer membrane of the entire cell. Plasma membrane of a cell. And the main composition is also different between plants and bacteria and fungi and the other organism that has a cell wall. So, for example, okay, the function of cell wall, I'm going to explain. It is to give the shape of the cell. So, meaning it maintains the shape of the cell to be in the structured form, and for mainly this purpose to give the structural support for the cell. Okay, so now there's one thing you must know, that cell wall exists in a different form for prokaryotics like bacteria, and for eukaryotic like if they mention plant cell. So for plant cell, they're made of cellulose, and if they're cell wall, and for bacteria cell wall, they're made of peptidoglycan. So that's the main composition. So for prokaryotic, the cell wall exists. Cell wall composition is peptido peptidoglycan. Okay, so the main composition is peptidoglycan for cell wall, but for eukaryotics, only if they mention plant cell, then it would have been correct, or unless then it's completely wrong because it does not exist in the animal cells, so cell wall is cancelled out. Okay, so now moving forward about the DNA in loops. Okay, so now this question is quite confusing because DNA loops can exist in many forms. It could be plasmid, it could be circular DNA, it could be anything. But we must use our logic over here. Okay, so now the thing is, if you reread the entire question, although you may not find exact thing about it, but you have to just use the logic that DNA in loops, what type of DNA in loops they're referring to, whether it's a plasmid or whether it's a circular DNA that the bacteria has its as a main form of genetic material. So what you must know about the DNA in loops is that the question is talking about main form of genetic material. Genetic material of the cell. So now, what we can do this is that we can easily cross this out. It is not common in both prokaryotic and eukaryotic. But there's a catch. Eukaryotic do have a very small circular DNA like mitochondrial DNA in animal and plant cells, and also chloroplast has their own genetic material because they are symbiotic organism. So they do have DNA in loops, but they're very smaller form. And also prokaryotics has lots of DNA in loops. Their primary genetic material is DNA in loops. Okay, so now if we compare DNA in loops. Okay, so DNA in loops exist in form of circular DNA in nucleoid nucleoid region of cytosol. Okay, so DNA in loops, if we're talking about prokaryotics, they're in the form of circular DNA. You know, the circular, it's also called circular chromosome, or you can say that. So that's the main genetic material of the prokaryotes. Okay, so wait, I'm going to mention in form of this. Okay, main genetic material. So I'm going to shorten in form of M. So main genetic material exists in form of circular DNA. So okay, so you must know that it's in form of circular DNA, and the nuclear region is the basically it's just the place where you might find the genetic material. But as I've mentioned, prokaryotics do not have any membrane-bound organelles, so it does not have any sort of nucleus that encloses this genetic material. So it just basically means the spot where you find this genetic material, that spot is called nuclear region in the cytosol. So it's just a free floating around the cytoplasm. Okay, now we will move forward about the eukaryotics. So eukaryotic, we must know the main genetic material. I'm going to short form in M. Okay, so main genetic material exists in form of linear chromosome. Linear chromosomes enclosed by nucleus. Okay, so in the eukaryotic and both animal and plant cells, main genetic material exists in form of linear chromosome enclosed by nucleus. Okay, so they're enclosed by nucleus and their main genetic material are linear chromosome, not a loop DNA. Okay, so we can cross this out. DNA in loops does not exist it's not common in both prokaryotics and eukaryotic. It's just common in prokaryotes only. Okay, so now moving on forward with option 4, cytoplasm containing ribosome. So we can easily mark this right. Okay, so the basic definition, what the ribosome is. So ribosome is basically a protein factory. Okay, it's a protein factory that synthesizes synthesizes uh, proteins. Okay, so it's a protein factory that synthesizes proteins, a polypeptide amino acid. So the main components that it uses it's mRNA. mRNA, it uses ribosomes, and it uses tRNA. Okay, so that's the three main components that are involved in the protein synthesis. And this protein synthesis, the main term is called translation. Okay, so this is the most important key point you must remember about ribosome. Whenever you hear the word ribosome, these are the three main things: protein factory, translation, and these main three components. Okay, so ribosome exists in both prokaryotics and eukaryotics in the cytoplasm because eukaryotic also have cytoplasm, prokaryotic also does. And they have ribosome in their cytoplasm. But the being said that ribosome exists in different forms in prokaryotic and eukaryotic animal cell, or it could be plant. In every eukaryotic, okay, so ribosome, now we're talking about the ribosome. Ribosome, so if you're talking about the ribosome in the prokaryotic organism, the ribosome exists in form of 70S ribosome. Okay, so this is a type of ribosome. And in the ribosome, we have a thing called subunit. So one is large subunit, one is a small subunit like this. So we have this subunits. So they enclose around the mRNA like that. So this is the large subunit, this is the small subunit. Okay, so similarly, so there's a name, there's a number for these subunits. So for prokaryotic, it is a 30S plus 50S subunit. Okay, so these are the subunit, and for eukaryotic animal cell, it is going to be 80S ribosome. Okay, 80S ribosome, and the main subunits are 40S plus 60S subunit. Okay, so these are the main subunits for 80S ribosome in eukaryotes and the subunits 30 plus 50S subunit for 70S ribosome in prokaryotes. So hence the option, this statement 4 is correct. It is common in both prokaryotics and eukaryotics. And one more thing I want to mention, the reason why they specified cytoplasm. The thing is that in animals, in the eukaryotic, the ribosomes can also be found in the structure called endoplasmic reticulum. So if the ribosome binds around the endoplasmic reticulum, it forms a rough endoplasmic reticulum. Okay, so basically rough endoplasmic reticulum, they receive the amino acids inside their lumen and then they transport it to the Golgi apparatus and do many other complicated functions. But prokaryotics has no such really complicated function. Their function are very simpler compared to eukaryotes. So they basically have ribosome free floating around the cytoplasm. And also eukaryotes also has free floating ribosome in the cytoplasm, but also they are found in the endoplasmic reticulum, which is around the nucleus, which is why it forms rough ER. Okay, so that's the thing about the question. So only the structure 1 glycogen and the structure 4 cytoplasm containing ribosome are both common in prokaryotics and eukaryotic animal cells. So hence the only correct option is option D, 1 and 4. Okay, so that's it about the question. So thank you guys for watching my video. I hope you find this video helpful. Please leave a like and subscribe to my channel. Thank you guys. Bye.